Hi everyone. In this video, we'll be looking at apoptosis. So we all know that apoptosis means programmed cell death, right? So this programmed cell death could be due to physiologic conditions, which is normal condition, or it could be due to pathologic conditions. Now let's see what goes into this process of apoptosis in detail, the mechanism of how it is initiated, what genes are responsible, and what happens in apoptosis that leads to cell death. Okay, so this process of apoptosis has two main pathways. It could happen in two main pathways. The first pathway would be your intrinsic pathway, also called as your mitochondrial pathway. And the pathway after this we'll be looking at is the extrinsic pathway, which is also called the death receptor pathway. Okay, most commonly every cell goes through apoptosis in the intrinsic pathway. Intrinsic pathway is the most common path chosen. Okay, so what happens in intrinsic pathway? Let's take this is our mitochondria. Okay, mitochondria has an inner membrane and an outer membrane. Remember, mitochondria is the powerhouse of the cell. So mitochondria has the electron transport chain in it that provides ATP for us to use, right? So in that electron transport chain, a major component is cytochrome C. And this cytochrome C is present inside your mitochondria because your electron transport chain is present inside your mitochondria and in between the membranes, right? But if this cytochrome C escapes the mitochondria and goes out to the cell, that could be lethal. That could lead to a series of reactions that can cause cell death. Okay, so how does this cytochrome C escape the cell when apoptosis is needed? Okay, so the mitochondrial membrane integrity is controlled by a gene called your BCL2 gene. Okay, so now depending on the situation, this BCL2 gene can produce multiple proteins. Those proteins can influence the cell to undergo apoptosis or not undergo apoptosis. So there are two kinds of protein, anti-apoptotic and pro-apoptotic. Anti-apoptotic protein are the ones that do not support apoptosis, whereas the pro-apoptotic protein are the ones that support apoptosis. So according to the situation, this BCL2 gene that is responsible for the membrane integrity of the mitochondria will produce your pro-apoptotic or anti-apoptotic protein. Now let's see what happens. In anti-apoptotic protein, this BCL2 gene produces these following ones: BCL2, BCLX and MCL1. And these contains BH1 to 4. So these are all gene names. You just have to remember that. Okay, they contain the BH1 to 4 domains in them, these three. So when these three proteins are produced, that means it's anti-apoptotic, right? So anti-apoptotic means there is no apoptosis. As a result, there will be no leakage of cytochrome C into the cytoplasm. We don't want that to happen. If cytochrome C leaks, then the cell will start undergoing apoptosis. So you need to prevent that because now you have anti-apoptotic proteins, right? So BCL2, BCLX and MCL1, when it's produced, that means the membrane integrity is complete and cytochrome C is still inside the mitochondrial membrane. Okay, but whereas if pro-apoptotic proteins are produced that stimulate apoptosis, like your BAX and BAC, which contain the BH1 to 3 domain, what happens? The membrane integrity of the mitochondrial membrane decreases. As a result, cytochrome C escapes the mitochondria and will activate apoptosis and cell death. Let's see how that happens. So remember, if BAX and BAC, what they do is they activate the membrane by oligomerizing with the mitochondrial membrane. And oligomerizing means what it's going to do, it's going to make the membrane more permeable. So these proteins are going to activate the mitochondrial membrane by oligomerizing it and making it even more permeable for cytochrome C to escape. So as a result, now it's more permeable, cytochrome C goes to your cytoplasm. Okay, now before we see how cytochrome C causes destruction of the cell, let me tell you another set of proteins that regulates this process. Okay, these proteins are called your regulated apoptosis initiators. You have BAD, BIM and BID, PUMA and NOXA. These have only the BH3 domain in them. Okay, remember BH1 to 4 domain is in anti-apoptotic, BH1 to 3 is in pro-apoptotic, only BH3 means these are regulatory proteins. Now what does these proteins do? In case of stress, okay, ER stress, endoplasmic reticulum stress, in case of DNA damage, in case of signals for survival is less, these apoptosis regulated proteins will get activated. And what they're going to do, they are going to inhibit anti-apoptotic proteins and promote your pro-apoptotic proteins. As a result, you will have activation, oligomerization, increase in membrane permeability, and cytochrome C goes to the cytoplasm from the mitochondrial cytoplasm. From the mitochondria, it goes to the cytoplasm of the cell, and hence activating cell death. Okay, so these regulated apoptosis initiators are just a control group. So if there is less survival signal, if there is DNA damage, if there's a lot of problems, stress, endoplasmic reticulum stress, and that's going to be activated, and that's going to inhibit anti-apoptosis and promote pro-apoptosis. Okay, that means it goes to release of cytochrome C into the cytoplasm. Okay, now once cytochrome C is into the cytoplasm, it goes and combines with a protein called as APAF1, apoptosis activating factor 1. That forms a complex called your apoptosome. Now this apoptosome binds to an enzyme called caspase 9 in the cytoplasm, gives autolytic cleavage to the caspase 9 and forms an active caspase 9. Okay, what does active caspase 9 do? It's going to go and activate other caspases, specifically caspase 3, 6. We'll be talking about it more. So what are the role of caspases? Caspases are enzymes that cleaves proteins and destroys the cell. Okay, so whatever comes in front of it, it's going to cleave it and it's going to destroy it. So this apoptosome is activating caspase 9 and other caspases. As a result, this enzyme is going to go cleave every single normal protein in the cell, hence leading to cell death. Okay, it's going to go and destroy, because cell is majorly made up of protein, right? The cytoskeleton, all the micromolecular structures in the cell is made up of protein. Now these caspases are going to go destruct these proteins, hence leading to apoptosis. Now see, why this is called programmed cell death? Because the cell only is sending signals through anti-apoptotic and pro-apoptotic factors for this process to happen. Okay, that's why it's called programmed cell death. So now the mitochondria also produces these proteins called SMAC and Diablo proteins. These SMAC and Diablo proteins inhibits the inhibitors of apoptosis. Usually there are inhibitors of apoptosis. Okay, that doesn't let apoptosis happen just like that, because we don't want apoptosis to happen just like that. It happens only during special conditions, right? Special situations. So usually this SMAC and Diablo protein is going to inhibit this inhibitors of apoptosis, and as a result you will have no apoptosis. But in worse conditions, this process also will be inhibited, and as a result you will have apoptosis. Okay, so just remember, SMAC and Diablo inhibits the inhibitors of apoptosis, and as a result there is no inhibition, instead there is apoptosis. Okay, now what about the apoptosis extrinsic path, the death receptor path? Okay, so this takes place in the plasma membrane, in the cytoplasm. So what happens here is the FAS binds to an FAS ligand. This FAS ligand is present in your cytotoxic lymph cells, cytotoxic lymphocytes, and in cells that recognize self-antigens. So once this FAS binds to FAS ligand, it forms a domain for FADD. Okay, now this FADD is going to convert procaspase 8 or 10 into active caspase 8 or 10. Now again, these active caspase 8 or 10 are going to go ahead and destroy your proteins across the cell and cause apoptosis. Now usually what happens during some viruses, they produce a protein called flip protein. That flip protein is going to inhibit this procaspase 8 or 10. As a result, it doesn't progress to apoptosis. So usually in cancers caused by virus and HIV, etc., what happens is you don't have cell death, right? You have excess number of cells in cancers. So that's how it's preventing apoptosis to happen. As a result, there is excess cells, and that is through this flip protein. Now also another important fact to know here is caspase 9, 8, 10 also promotes the activation of caspase 3, 6, which cleaves the DNA and fragments the nucleus. As a result, there is cell death. Now finally, once everything is cleaved and destroyed, where does all this go? Phagocytosis happens. The phagocytes come and consume all this. And when phagocytes come and consume these apoptotic bodies, that process is called efferocytosis. When phagocytes come and consume the apoptotic bodies formed by the cleavage due to caspase, that is called efferocytosis. And how does this phagocyte get to know that this is the body that we should consume? Because that body will be coated with phosphatidylserine or complement 1 protein, C1q protein. Okay, so once that is coated with these proteins, the apoptotic bodies, the phagocytes will come and phagocytose the apoptotic bodies, and hence it will destroy the entire apoptotic body. Okay, So this is apoptosis in a nutshell, two main pathways, intrinsic and extrinsic. I hope you understood the mechanism behind what really happens during apoptosis. Thank you.